Hi, my name's Natalie Trevaskis. I'm head of the Lymphatic Biology and Drug Delivery Research Laboratory at the Monash Institute of Pharmaceutical Sciences at Monash University, Melbourne, Australia.
Hi, I'm Gracia. I'm a postdoctoral researcher at Monash Institute of Pharmaceutical Science, Monash University.
In this video, we'll give you a brief introduction to our recent paper published in the American Journal of Physiology, Gastrointestinal and Liver Physiology. The paper is titled Organ-Specific Lymphatics Play Distinct Roles in Regulating HDL Trafficking and Composition. We hope this brief introduction will entice you to read our paper.
Lipoproteins are complex particles made of different lipids and proteins, which are produced by our body to enable transport of lipids between different organs and tissues. Disruption in the assembly, transport, and metabolism of lipoproteins has been associated with increased risk of cardiometabolic diseases such as atherosclerosis and fatty liver disease. Lymphatic vessels drain the main organs that produce or metabolize lipoproteins such as the small intestine and liver. Mesenteric lymphatics draining the intestine and hepatic lymphatics draining the liver converge at the thoracic lymph duct, which collects approximately 80% of lymph in the body and empties into the blood circulation. The mesenteric lymphatics transport dietary lipids in the form of chylomicrons and VLDL, whereas hepatic lymphatics have been shown to transport VLDL secreted by the liver. Recent studies have further revealed that peripheral lymphatic vessels are a conduit for HDL transport from peripheral tissues back to the blood circulation during the process of reverse cholesterol transport. This function was vital near the heart, where lymph-mediated HDL transport from artery walls reduced atherosclerotic plaque size. However, there remain significant gaps in our knowledge of the trafficking of different lipoprotein types by lymphatics in different organs, such as the role of mesenteric and hepatic lymphatics in HDL transport. We therefore aim to determine the contribution of mesenteric and hepatic lymph transport to total lymph lipoprotein and HDL transport, which is represented by thoracic lymph. Secondly, we aim to investigate whether the HDL in lymph are organ-specific in composition and are modified during trafficking through lymph. To test our hypothesis, we cannulated the mesenteric, hepatic, or thoracic lymph duct of anesthetized non-fasted rats to collect lymph fluid. Using ultracentrifugation, lymph fluid was separated into lipoprotein fractions, and the HDL fractions were isolated for protein and lipid composition analysis. We found that mesenteric lymph and, to a lesser extent, hepatic lymph contribute the majority of thoracic lymph fluid and lipoprotein-associated protein and lipids. Interestingly, while the HDL-associated protein and lipids of thoracic lymph HDL were mostly derived from the mesenteric lymph, HDL-associated cholesterol was equally derived from the mesenteric and hepatic lymph. We further found that the HDL in the three lymph sources were compositionally distinct and also different from plasma HDL. This confirms that lymph HDL species are organ-specific and suggests that HDL from the intestine and liver may drain directly into the mesenteric and hepatic lymphatics and be subsequently modified during passage to the thoracic lymph duct.
Overall, these studies contribute to an enhanced understanding of high-density lipoprotein trafficking and composition in the lymphatics. In particular, these studies identify that high-density lipoproteins may be modified during passage through the lymphatics, and these processes may modify the biological properties and functions of high-density lipoproteins and their impact on cardiometabolic disease. Future studies may seek to identify the mechanisms that drive high-density lipoprotein modification within the lymphatics.